Hi, I'm Ryan O'Mara, a PhD student from the University of Ottawa and Ottawa Hospital Research Institute. This protocol will describe methods on how to produce enriched populations of oligodendrocyte precursor cells from mice, which may be differentiated alone or in combination with mouse dorsal root ganglion neurons to produce myelinating co-cultures. Let's begin. To give a brief overview of the procedure, neonatal mouse cortices are dissociated and seeded into T25 flasks. After a culture period of 9 days, an astrocyte monolayer forms on the base of the flask, on top of which oligodendrocyte precursor cells are proliferating. Oligodendrocyte precursor cells are then obtained and purified and can be subsequently differentiated into mature oligodendrocytes in an enriched culture. If myelinating co-cultures are desired, dorsal root ganglion neurons from P5 to P10 mice are cultured alongside the mixed glial cultures. Over a period of 9 days, these neurons form a dense neurite bed on which oligodendrocyte precursor cells may be seeded. This allows for oligodendrocyte-mediated wrapping of the neurites, thus resulting in a myelinating co-culture. First, whole brains from P0 to P2 mice are placed in ice-cold MEM and transferred to a dissection microscope. Using a scalpel, cut through the meningeal layer of each cortex in a longitudinal manner. Then pry off the meninges from the underlying cortex. Next,